Module 27 Phylum Nemetohelminthes. Phylum Nemetohelminthes. Learning objective. After reading this module, the learner will understand the following: importance of nematode in animal and man, ideal environmental for development of larval stage nematode, various pattern of nematode life cycle and its stage, control of larval stage. Introduction. Nematodes. Nematodes or roundworm are very important group of helminths which infect animal and human. The nematodes have both direct and indirect life cycle affecting wide range of animal. Nematodes are free-living or parasitic and unsegmented cylindrical in shape. Nematode cannot be stand for identification. Some of the nematodes have on their cuticle specialized adhesive structure such as hooks in Tetrameres species, cephalic vesicle in Esophagostomum species. Some of the nematodes have lateral flat circular expansion called as alley. If the alley is situated in the cervical region, it is called as cervical alley, and in posterior region as caudal alley. Alimentary canal is present. They have a body cavity or coelom, but it is not true coelom, pseudocoelom. The extremities are tapering, except the female of Tetrameres species, which become almost spherical after occupational. The sexes are separated. The body is covered with cuticle. The cuticle is provided with circular annulation, or it may be smooth, or it may have longitudinal striations. The cuticle is relatively thick in nature and may extend into the buccal capsule, esophagus, rectum, and distal portion of genital duct. Sometimes the cuticle forms a special adhesive structure, for example, hooks in Tetrameres species, male simple or complicated cuticle thickening, or in Gongylonema species, cervical color or cephalic color in Physaloptera species, cephalic vesicle in Esophagostomum species. Cuticle is formed by underlying subcuticle layer or muscular layer. The subcuticular layer is called as hypodermis. In free living form, the hypodermis is composed of number of cells, whereas in parasite form, hypodermis consists of syncytium with a number of nuclei. Subcuticular layer form a four longitudinal thickening on the inner aspect, which are situated on dorsal, ventral, and lateral aspect. Muscular layer situated next to subcuticular layer and is divided into four quarters by longitudinal thickening. Digestive system of nematodes. Digestive system of nematodes. Mouth is situated at anterior extremity. Sometimes it may be subdorsal or subventral. Mouth is surrounded by lips in ascarid. The mouth is surrounded by three lips on one dorsal and two lateral, and each lip possesses two papillae. The spirulate. The mouth is surrounded by two lips. Each lip possesses three papillae and subdivided into three parts. In the members of Strongylidae, the lips are absent. Instead of lips, the secondary structure may be developed, known as lip crown. It consists of number of pointed processes. The lip crown arises from the rim of the mouth, called as external lip crown. Those lip crown arising from brim of the buccal capsule is known as internal lip crown. Example: Esophagostomum species. In some species, the buccal capsule has a thick cuticular wall or teeth-like structure. Example: Encyclostomum species. Esophagus. The structure of esophagus is very important character for identification of species. It is a strongly muscular organ which tri-radiated lumen. The lumen is covered with thick cuticular lining and has three glands secreting digestive enzymes. At the posterior end of esophagus, a bular swelling may occur in some species, and it is known as esophageal bulb, which contain valvular apparatus. Example: Heterachus gallinarum. Type of esophagus: rhabdiform, filiariform. Rhabdiform esophagus, club shape anterior portion is connected by a narrow neck to pear-shaped posterior bulb. This type occurs in the first larval stage of nematodes and adult from the non-parasitic generation of adult worm Reptidida. Filiariform esophagus. Filiariform esophagus. Esophagus is club shaped without posterior bulb. It occurs in the second and third larval stage of nematode and also in parasitic generation of Reptidida. Excretory system of nematodes. Excretory system is not carried out by phloem cell as in the case of trematodes. Excretory regulation by osmotic regulation. This system consists of a pair of unbranched lateral vessels embedded in hypodermis which open through excretory pore. Excretory pore is situated short distance behind the anterior extremity. Nervous system of nematode. Nervous system consists of number of ganglia connected by nerve fiber, forming the nerve ring around the esophagus from the central nerve ring. Six nerve trunk run anteriorly and six trunk to posterior part. Class Nematoda. Class Nematoda. Sense organs. Amphid. Amphid consists of a pair of depressions on each side or head and well supplied by nerve fiber. The amphid are situated either lateral or posterior lateral aspect of head end. The function is chemoreceptor. Plasmid. A pair of depression occurs in each side of tail end. Earlier, the plasmid were considered as important characters for identification. Nematode with plasmid come under plasmidia and without plasmid come under aplasmidia. Cervical papillae. Example: Esophagostomum species. A pair of papillae occurs in the cervical region. Papillae in the middle of body. Genital papillae. In female, a pair of papillae occurs in vulvar region. Reproductive system of nematodes. Sexes are usually separate. Sexual dimorphism occur. Usually male worms are smaller than the female. Male reproductive system of nematodes. It consists of single testis, wide difference seminal vesicle, and ejaculatory duct. This ejaculatory duct open into the cloaca. In most of the species have one or two spicules. Spicules are like in the sheath which open into the cloaca. Spicules are chitinized, pigmented. They vary in size and shape. The number, size, and shape is very important character for identification of species. Function of spicules: for attachment during copulation and expand the vagina for direct the flow of sperm. In many species, the wall of the cloaca is provided with cuticular thickening. This thickening assists the spicule movement. Cuticular thickening on the dorsal wall of cloaca is called as gubernaculum. Cuticular thickening on the ventral wall of cloaca is called as telamon. Female reproductive system of nematodes. Female reproductive system of nematodes. It consists of a pair of ovary, oviduct, and uterus, which end in a short vagina and open at vulva. In some species, like Homunculus contractus. The vulva is covered with vulval flap of nerve. At the junction of uterus and vagina, a small muscular organ may occur and it is known as ovigesctor. Its function is assisting in laying eggs. One female may lay several thousand eggs per day. Eggs of nematode. Nematodes are oviparous or viviparous oviparous nematodes that lay eggs which contain only yolk material when laid. Example ascarid. Oviparous nematode they lay eggs. It contains fully developed larvae when laid. Example Suburula brumpati. Viviparous giving birth to larvae. Example Filariid nematode. The larvae of Filariid nematode is called as microfilaria. Nematode eggs vary greatly in shape and size. Usually they are round, oval and ellipsoidal in shape and covered with three layers. In some species the middle layer is interrupted with operculum or plug. The plug may be occurs at one end or at both the end of egg. Example plug at one end. Oxyuris equi plug at both ends. Trichuris ovis. Development and hatching of nematode egg. Development and hatching of nematode egg. Original egg cell divided into 2, 4, 8, 16, 32 and so on. The developmental stage consists of morula and tadpole stage. After completion of larval development, the eggs are ready to hatch. Hatching of egg depend on the species of worm. Egg may hatch outside the host in the environment or may hatch after ingested by D or H or IH. Example in ascarid, the egg hatch only after ingestion by the D or H. In Spirocerca lupi, the egg hatch only after ingestion by the IH. In strongyle species, the egg hatch in the environmental dead so outside the host. Hatching of the egg in the environment is controlled by temperature, moisture, larval movement, and enzymes secreted by larva. Hatching of egg inside the host is controlled by host stimuli like CO2 concentration. Biomix of larvae, usually for months or ecdysis L1, L2, L3, L4 occurs in nematode. During molting, the old cuticle is replaced by new one. The L1 and L2 feed on bacteria. L3 cannot feed because the cuticle of L2 retains as a protective sheath around L3 larvae infective stage. In most of the nematode species, L3 is the infective stage whereas in ascarid L2 is considered as a infective stage. Life cycle pattern of nematode. Life cycle pattern of nematode. Direct life cycle. Egg develop and hatch outside the host. The larvae live as a free living for some time and later become infective. The infective larvae enter the host by two ways. In some species, the infective stage enter into the host through mouth along with feed and water. Example: Strongylidae and Trichostrongylidae. But the hookworm larvae infective stage enter into the host by skin penetration. Example: Encyclostomum and Buenostomum species. The development of egg occur in the environment, but eggs do not hatch outside the host in the environment. Host getting infection by ingestion of embryonated egg. Example: Ascaris. Indirect life cycle. Indirect life cycle. Egg hatch outside the host or ovoviviparous. The larvae are free living for some time, then enter into the suitable host and become infective stage. Final host acquired infection by ingestion of infected host or infective stage injection by the host while feeding. Example Metastrongylidae and Herbronema. Eggs do not hatch outside the host. They hatch only after ingestion by host. Acquired infection by injection of host. Example Spirocerca lupi. Viviparous in which the adult female worm directly give birth to larvae that enter into the blood circulation of the host. From the blood, it is taken up by blood sucking host. Within the host, the larvae become infective. The infection of final host by biting of infected host by inoculation. Example Cyteria digitata. Classification. The class Nemetoda is classified as below. Class Nemetoda, order Ascarida, superfamily Ascarididae, family Ascaridae, genera Ascaris, Parascaris, Toxocara, superfamily Oxyuridae, family Oxyuridae, genera Oxyuris and Robias, superfamily Subuloroidae, family Hetrachidae, genera Hetrachis, Ascaridae, family Subularidae, genus Subulera, order Rhaptidida, superfamily Rhaptidoidae, family Strongyloidae, genus Strongyloids, order Strongyloida, superfamily Strongyloid, family Strongylidae, genera Strongylus, Triodontophores, Craterostomum, family Trichonematidae, genera Cytostomum, Silicolicus, Silicodontophorus, Silicostephanus, Posteriostomum, Glycocephalus, Burgellatia, Coniagenum, family Dracunculidae. Thank you.